Usually when we talk about a medical diagnosis, there's this expectation of like extreme, almost mechanical precision.
Right, yeah. Like it's a binary system.
Exactly. You break your arm, right? And the x-ray shows that jagged white line on the dark film, and the doctor just points and says, well, there it is. It's incredibly straightforward.
And as humans, we find an immense amount of comfort in that kind of diagnostic clarity. We want things to be visible. We want them to be perfectly categorized.
We really do. But, the moment you step into the world of molecular biology and genetics, that X-ray machine just completely shatters.
Oh, that's what...
You're suddenly looking at a landscape that it's profoundly murky. You aren't looking at static bones anymore. You are looking at a microscopic turf war where DNA basically writes its own rules on the fly, constantly adapting, replicating, and this is the crazy part, occasionally making massive consequential errors.
Yeah, it really is the antithesis of that clean medical diagnosis. But what's fascinating, at least to me, is that beneath the apparent chaos of a cell, there is this profound, almost terrifying mathematical elegance.
And that invisible architecture is exactly what we are exploring with you today. We're going to examine a really fascinating stack of research that explores how genetic information is controlled, restricted, and copied.
Which is the foundation of everything, really.
Right. So we are starting small, looking at some highly detailed molecular biology texts detailing the cutthroat world of bacteria. Specifically, there's this dynamic called plasmid incompatibility,
which at its core is just a study in absolute microscopic perfection.
Yeah. And then from that microscopic perfection, we are going to zoom way out. We have this incredible evolutionary biology paper by Michael W. Nachman and Susan L. Crowell that calculates the exact mutation rate of the human genome.
It's a brilliant paper.
It is. We're going to see what happens when the DNA copying process isn't so perfect and what it actually means for your own survival over millions of years of evolution.
So it's two vastly different scales of life, but they fundamentally ask the exact same question, which is, how does DNA regulate itself? And the contrast between how a single-celled bacterium handles that regulation versus how a complex human being handles it, well, it's staggering.
Okay, let's unpack this. Let's start on the microscopic side. I was looking through the notes on bacterial plasmids, and the way the texts describe them, they sound almost like rogue elements.
That's a good way to put it, yeah.
They are these extra chromosomal circular pieces of DNA. They exist inside a bacterium, but they are entirely separate from the bacterium's main chromosome, right? And they replicate completely on their own schedule.
They are entirely autonomous, yes. And the critical distinction to understand here, and this is really important, is that under normal, everyday conditions, a bacterium does not actually need a plasmid to survive.
Wait, really? It's just extra?
Yeah, completely extra. The main bacterial chromosome handles all the essential housekeeping genes. So, the proteins for metabolism, the structural components of the cell wall, basically everything required to just keep the lights on.
So the plasmids are basically non-essential, but they aren't just freeloaders, right? Because they stick around for a reason. They carry advantageous traits. And the classic example that just keeps coming up in the reading is antibiotic resistance.
Exactly. They act as an evolutionary insurance policy.
Okay.
If a bacterium is just swimming along in a normal environment, the plasmid is just, extra baggage. But if that environment suddenly floods with a lethal antibiotic like penicillin, well, that non-essential plasmid, which happens to carry a gene that destroys penicillin, suddenly becomes the sole reason the bacterium survives.
Right. But that insurance policy has a massive premium attached to it. The bacterium doesn't just get to hold on to this plasmid for free. It has to expend a tremendous amount of cellular energy, right? Like gathering nucleotide building blocks, using up ATP just to constantly copy this extra piece of DNA.
It's extremely expensive for the cell. And that energy cost creates a very dangerous tightrope walk for the plasmid itself. Which brings us to a fundamental concept in molecular biology called copy number.
Copy number.
Yeah, the copy number is essentially the fixed characteristic number of copies of a specific plasmid that exist inside a single bacterial cell under constant conditions.
Meaning some plasmids might only maintain 1 or 2 copies of themselves per cell, while others might maintain, 50 or 100.
Exactly.
And the plasmid itself carries the genetic code that dictates this number, right? It controls its own replication frequency.
It does. But it's not just about making the copies. It's about what happens when the bacterial cell eventually divides.
Right, because if a bacterium splits into 2 daughter cells and all 50 copies of the plasmid happen to be floating on the left side of the cell, the daughter cell on the right gets nothing. It's just out of luck.
Exactly. And to prevent that, plasmids have evolved what is known as a partitioning system. The partitioning system ensures that during cell division, the replicated plasmids are physically distributed so that each new daughter cell receives at least 1 copy.
Okay.
If that system fails, the plasmid is lost to the next generation, which means it basically goes extinct in that particular lineage.
So I want to try an analogy here to sort of visualize this dynamic. Imagine the bacterial cell is like a manufacturing factory.
Okay, I like where this is going.
The main chromosome is the factory manager, and it's handling all the essential day-to-day operations, keeping the assembly line moving, paying the electric bill, that kind of thing. Plasmids are like freelance contractors who walk into this factory.
Right.
They bring their own highly specialized blueprints, maybe a blueprint for how to dismantle a toxic chemical that might leak into the factory.
Yeah, they bring specialized situational value. That's a great analogy.
But here's what I don't quite grasp from the texts. If these plasmids are autonomous freelancers and they literally control their own copying mechanisms, what is stopping them from just running over to the factory's printing press, printing 10,000 copies of their blueprint, taking up all the factory floor space, and effectively just taking over?
The short answer is self-preservation.
Self-preservation.
Yeah. A plasmid's copy number is strictly, and I mean mercilessly, regulated by its own replication control system because over-replication is a death sentence.
Oh, because it would kill the host.
Precisely. If a plasmid were to replicate without limits, it would monopolize the host cell's metabolic resources. It would drain all the ATP, exhaust the nucleotide pool, and the bacterium, the factory in your analogy, would die.
And if the factory burns down, the freelancer dies inside it.
Right. They are obligate parasites in that sense. They have to perfectly balance their own selfish evolutionary drive to replicate with the physiological limits of their host. And to your earlier point about cell division, the mechanisms for initiating replication and the physical mechanisms for partitioning are completely independent of one another.
Meaning they are 2 totally separate systems.
Yes. A plasmid has one highly tuned system for deciding when to make another copy, and a totally distinct mechanical system for ensuring 1 copy goes to the left pole of the cell and 1 goes to the right during division.
This level of microscopic regulation is mind-blowing, but I guess, why should you, the listener, care about how a bacterial freelancer behaves?
Well, because it's the basis of so much technology.
Right, because this specific tightrope regulation is the absolute foundation of all modern genetic engineering. Like when scientists create synthetic insulin for diabetics, or when they engineer bacteria to consume ocean oil spills, they are using plasmids to deliver the instructions.
Exactly. If we don't understand how copy number and partitioning work, the engineered factory just collapses.
And that highly tuned system is incredibly fragile when you introduce competition, isn't it?
Extremely fragile.
Which brings us to the concept of the turf war. The literature calls this plasmid incompatibility, and I read the definition, and it sounds almost like a microscopic roommate dispute that ends in an eviction.
That's actually not a bad way to describe it at all. In molecular terms, plasmid incompatibility is the inability of 2 different plasmids to coexist stably within the same bacterial cell line over successive generations. Assuming, of course, there is no external selection pressure forcing the cell to keep both.
Let's clarify external selection pressure real quick. That just means we aren't artificially flooding the petri dish with 2 different antibiotics, right?
Correct.
Because if we did that, the cell would be forced to keep both plasmids just to survive the poison. But if we let them grow normally in a safe environment, they are going to compete.
Right. Without that gun to their head, they are subject to internal cellular dynamics. And the general rule observed by microbiologists is fascinating. Closely related plasmids tend to be incompatible with each other, while distantly related plasmids tend to be perfectly compatible and can coexist peacefully.
Okay, I have to stop you there because when I was reading the texts on this, it completely broke my logic.
How so?
Well, the text explicitly says that if 2 plasmids are genetically identical, literally the exact same sequence, they are completely compatible. They coexist fine.
Yes.
But if 2 plasmids are genetically distinguishable, meaning they carry different traits, yet they share the exact same replication mechanism, they're incompatible. Why wouldn't sharing a mechanism make them compatible? If they speak the same language, shouldn't they get along better?
I know, it is deeply counterintuitive at first glance.
Yeah.
But you have to stop thinking about them as conscious roommates and start thinking about how blind the cellular machinery actually is.
Blind.
It all comes down to a shared pool of resources and pure statistical probability. So let's walk through the scenario of the identical plasmids first.
Okay, 2 exact copies of the same plasmid.
Let's say this specific plasmid's control system is mathematically tuned to maintain a total of 10 copies within the cell. It uses specific replication proteins. We'll call them rep proteins.
Got it.
If you have 2 identical plasmids, plasmid A and plasmid A prime, they're both drawing from the same pool of rep proteins. The cellular machinery cannot tell them apart. It just counts the total repressor signals and says, okay, we have 10 copies total, stop replicating.
So it doesn't care who is who.
Right. It doesn't matter if you have 5 of A and 5 of A prime or 8 of A and 2 of A prime. Because they are genetically identical, the cell still functionally has 10 copies of the exact same genetic blueprint. The cell functions normally.
The end result for the bacterium is identical no matter what the ratio is.
Exactly. But now, change the scenario. Imagine 2 plasmids that are slightly different genetically. Let's say plasmid A carries resistance to ampicillin. Plasmid B carries resistance to tetracycline.
Okay, different traits.
Different traits. But crucially, they evolved from the same ancestor. So they still rely on the exact same replicon, the exact same replication control system.
So they are throwing the exact same stop-and-go signals into the cell, competing for the exact same rep proteins.
Yes. And the cell machinery is completely blind to the fact that they carry different antibiotic resistance genes. It only sees their replication signals. So the cell still just tries to maintain a total of 10 copies between the 2 of them.
Okay, I think I see where this is going.
Now, let's say a cell starts with 5 copies of plasmid A and 5 of plasmid B. In the next generation, they replicate. By pure random statistical fluctuation, plasmid A might replicate faster and hit 6 copies, while plasmid B only hits 4.
Because there's no system ensuring a perfect 50-50 split. The cell just stops everything once the total hits 10.
Right. And then that cell divides. And the next generation starts with a skewed ratio. Maybe it goes to 7 and 3, then 8 and 2. Eventually, over successive generations of the bacteria dividing and the plasmids randomly competing for that capped pool of 10 slots, one of them will randomly hit 0.
Wow.
And in biology, once you hit 0, you don't come back.
That makes perfect sense now. It's like having a joint bank account with 2 people who both have identical debit cards, but they aren't communicating with each other.
I love that analogy.
The cell doesn't care who spends the money. It just shuts off the cards when the balance hits 0. One person might accidentally spend all the funds, leaving the other one with absolutely nothing.
That is a brilliant way to frame it. And this probability dynamic creates 2 distinct types of incompatibility: symmetric and vectorial.
Symmetric incompatibility is basically a fair fight.
Yes, it means both co-resident plasmids have an equal, random chance of being the one that eventually gets lost. It's a coin toss that plays out over generations.
And vectorial incompatibility means the game is rigged from the start.
Exactly. In vectorial incompatibility, one plasmid is lost almost exclusively. This happens when one of the plasmids has a competitive advantage. For example, some plasmids are incredibly flexible and possess a second independent replicon.
Meaning they have 2 ways to copy themselves.
Right. They have 2 different ways to initiate replication, while the competing plasmid only has 1. The flexible plasmid will almost always outcompete the rigid one, monopolize the machinery, and drive the other to extinction.
But they don't just fight over the replication machinery, right? There is a whole separate battlefield when it comes time for the cell to divide. The texts call this segregational incompatibility.
Remember that partitioning system we discussed? The mechanism that ensures plasmids get physically pulled to the opposite poles of the cell before it splits in half.
Yeah.
That system relies on very specific proteins, usually called para and par, which bind to a specific DNA sequence on the plasmid called the para site.
So if 2 totally different plasmids happen to share the exact same para sequence, they start competing for the same para and par-B proteins.
And it causes absolute chaos for the cellular machinery. The machinery is supposed to pair up 2 identical plasmids and pull them apart. But if plasmid A and plasmid B have the same par site, the par proteins might accidentally pair plasmid A with plasmid B.
So instead of separating them, it accidentally walks both of them into the exact same daughter cell, leaving the other daughter cell completely empty of one of the plasmids.
which is an incredibly inefficient way to segregate. Over time, this constant misdirection leads to one of the plasmids failing to be inherited across the population, and it simply fades out of existence in that bacterial lineage.
Okay, so we've established that plasmids are strictly controlling their copy numbers to survive, and if they share control systems, they engage in this statistical fight to the death.
Right.
But I want to get into the physical mechanics of this. How are they actually physically stopping themselves from copying? Because it's not like there's a little brain in there making decisions.
No, there is no brain. It is purely driven by thermodynamics and molecular interactions. And one of the most elegant mechanisms for this is direct control through something called antisense RNA.
The sources focus heavily on the E. coli, coli-1 type plasmids for this. Let's break down how a coli-1 plasmid actually starts copying itself, because the braking system won't make sense without understanding the gas pedal.
Fair point. For a coli-1 plasmid to initiate DNA replication, it has to first transcribe a relatively long precursor RNA molecule. The texts refer to this as RNA2. It's about 700 bases long.
So the plasmid is reading its own DNA and printing out this 700-letter-long strand of RNA2. But RNA2 isn't the final key, right? It has to change its shape.
Right. Single-stranded RNA is very flexible. In order to kick off the DNA copying process, the 3' end, one specific tail of this RNA2 strand has to fold back on itself and form a very specific 3D secondary structure. It forms what we call a stem loop.
And when it forms that specific origami shape, it can physically slot into the DNA template at the origin of replication.
We call that process coupling. The RNA2 hybridizes with the DNA. Once that coupling occurs, an enzyme present in the host cell called RNase H comes along.
RNase H.
Yeah. RNase H acts like a highly specific pair of molecular scissors. It recognizes that RNA-DNA hybrid, and it cleaves the RNA2 strand, leaving behind a free 3' hydroxyl chemical group.
And that free hydroxyl group is the starter pistol.
It is the absolute non-negotiable requirement for DNA polymerase, the heavy machinery that actually copies the DNA to swoop in and start synthesizing the new plasmid. Without that cleaved RNA primer, the polymerase can't do anything.
Okay, so RNA2 is transcribed, folds into a loop, couples with the DNA, gets cut, and boom, the plasmid copies itself. So how does the plasmid stop this process when it already has enough copies?
It uses a second molecule called antisense RNA. Specifically, a much shorter, a 108 base transcript called RNAi.
And what's brilliant is where RNAi comes from. It is transcribed from the exact same region of DNA as RNO2, but it's transcribed from the opposite strand, running in the exact opposite direction.
Which means RNAi is perfectly mathematically complementary to the 5' end of RNA2. If RNA2 is the lock, RNAi is the mirror image key.
So how do they interact in the physical space of the cell? Like what happens when they bump into each other?
They both have these tiny single-stranded loops at their ends. As they float through the cellular fluid, if an RNAi molecule bumps into an RNA2 molecule, their loops interact in a highly unstable, transient way. Biologists actually named this initial interaction the kissing complex.
The kissing complex. It's a very evocative name for a molecular collision.
It is, yeah. But that kiss is just the beginning. Once those loops interact, it triggers a progressive zipper-like pairing along the entire length of the RNAi molecule. It completely binds to the RNA2 strand.
And if RNA2 is bound up by RNAi, it is physically blocked from forming that necessary stem loop structure we talked about.
Exactly.
It can't couple with the DNA. RNA has nothing to cut. DNA polymerase has no primer. The entire replication process is stopped dead in its tracks.
It's a pure negative feedback loop.
The plasmids are basically constantly shouting, "Stop it," themselves.
Exactly. And the mathematics of it are beautiful in their simplicity. The regulatory RNAi is what we call constitutively transcribed. That means it is constantly being produced all the time without needing a trigger.
So the concentration of RNA floating around in the cell is a direct physical proxy for the number of plasmids present.
Yes. If you have 2 plasmids, you have a little bit of RNAi. If you have 10 plasmids, you have 5 times as much RNA. The higher the concentration of RNAi, the higher the statistical probability that it will intercept and kiss a newly forming RNA toy or primer before it have the chance to initiate replication. The plasmids build their own braking system, and the more plasmids there are, the harder the brakes are applied.
That is stunning. Oh, and the sources also mention a specific protein involved in this coli-1 system called ROM, or sometimes ROPE. What is its role if the RNA is already doing the blocking?
ROM is a small 63 amino acid dimeric protein. Surprisingly, it's actually not essential for the plasmid to replicate or even to maintain its copy number.
Then what does it do?
It acts as a molecular matchmaker. It physically binds to both RNAi and RNA2 simultaneously. right during that initial unstable kissing phase.
So it grabs them both and holds them together. It's like molecular duct tape.
That's a perfect visualization. It stabilizes that transient kissing complex about 100-fold. By making the binding that much more efficient and rapid, it severely enhances the incompatibility effect we discussed earlier, and it actually decreases the overall copy number of the plasmid by a factor of 2 to 5. It tightens the tolerance of the whole system.
Okay, so that is how COL1 works, by physically intercepting the RNA primer. But the sources also dive into a different group of plasmids, the INCA type plasmids, which includes R1 and R65. They also use antisense RNA, but they deploy it in a completely different way.
Right. While COL1 operates at the level of the primer, inhibiting the actual initiation of replication, INCA plasmids target the process of translation. They stop a critical protein from ever being built.
This critical protein is called replication. The texts note it's cis-acting, meaning it generally only acts on the DNA molecule that produced it, and it is the absolute rate-limiting factor for these plasmids. If the cell has replication, the plasmid replicates. If it doesn't, it waits.
So the entire survival of the INCA plasmid hinges on carefully controlling how much replication protein is synthesized. To do this, the plasmid has two separate promoters, basically two starting lines for the replication gene. There is an upstream promoter that is constantly active, churning out a baseline level of transcripts, and then there is a downstream promoter that is heavily regulated by a repressor protein called copy.
Copy acts like a night watchman. Under normal conditions, when the plasmid has reached its target copy number, copy sits squarely on that downstream promoter, physically blocking it from being used.
And we know how powerful this watchman is because the sources cite an experiment where researchers intentionally deleted the copy gene from the plasmid.
What happened?
Without that repression, the plasmid copy number violently jumped by 8 to 10-fold. It shows how vital that transcriptional repression is.
But copy is just the course adjustment, right? The real finesse, the fine-tuning, happens post-transcriptionally with an antisense RNA called COPA.
COPA is a small 90-base antisense RNA. Just like the COL1 system, it is transcribed in the opposite direction of the replication messenger RNA. Its target is a specific leader region on the replication mRNA called COPT.
So COPA is constantly being produced. It floats over to the replication messenger RNA and it binds directly to the COPT region, forming a double-stranded RNA duplex.
And here is the physical roadblock. When COPA binds to COPT, it physically obstructs the ribosome. The ribosome is the cellular machine that reads mRNA and translates it into a protein. If the ribosome hits that double-stranded roadblock, it can't read the code.
No translation means no replication protein. No replication protein means no replication.
And again, the elegance is in the concentration. COPA is synthesized continuously. Its abundance in the cell is strictly proportional to the number of plasmids. It's a highly dynamic, continuous braking system. If the cell grows and the copy number effectively dilutes, the concentration of COPA drops. The brakes are momentarily released, the ribosome translates replication, and the plasmid quickly replicates to recover its numbers.
It's incredible how mechanical it is. It's not abstract biology. It's literal pieces of molecular tape sticking to each other to stop a gear from turning. But antisense RNA isn't the only way plasmids achieve this, is it? Because that brings us to the other major mechanism in the sources, interons and handcuffing.
Yes, this represents a completely different philosophy of regulation. This is a mechanism of indirect control. Plasmids like PSC101, the F plasmid in R6K, don't rely on antisense RNA transcripts. Instead, they utilize specialized structural regions at their origin of replication called interons.
An interon is essentially a cluster of direct DNA repeats, just a short, specific sequence of DNA letters repeated over and over in a row. And these interons require a specific transacting replication and initiation protein to function. In the case of the R6K plasmid, it's called the pi protein.
For a very long time in molecular biology, the prevailing theory for how interons controlled copy number was known as the titration model.
I read through the titration model, and it views interons basically as molecular sponges. The idea is that the cell produces a limited, steady amount of the rep protein, like the pi protein. To initiate DNA copying, these pi proteins have to bind to the interons at the plasmid's origin.
So, following the logic of the titration model, when a plasmid replicates, you suddenly go from having one plasmid to having two, which means you suddenly have twice as many interon sponges in the cell.
These fresh, empty iterons immediately soak up all the free pi proteins floating in the cellular fluid. They titrate the protein out of the solution, dropping the overall concentration of free pi proteins below the threshold required to trigger another round of replication.
It's an incredibly logical, intuitive model. It makes perfect sense on paper. The problem is, well, when researchers actually tested it, it fell apart.
Right. The texts describe an experiment that totally dismantled the titration model. Researchers figured if this is purely a matter of soaking up proteins, what happens if we overwhelm the sponges?
That is the ultimate stress test for the theory. If you artificially pump a massive, overwhelming amount of rep protein into the cell, the iterons should become completely saturated. The concentration of free rep protein would remain sky high, and it should trigger uncontrolled runaway replication of the plasmid.
But the researchers did exactly that with the R6K plasmid. They engineered the cell to overproduce the pi protein up to 10,000 times the amount normally required to saturate the iterons. 10,000 times. And the plasmid copy number barely budged. It did not increase.
Which is a brilliant moment in science. It proves that the simple titration model was missing a massive piece of the puzzle. If the sponges are overflowing, why is the plasmid still refusing to replicate?
Enter the coupling or handcuffing model. This completely changed how I visualize DNA inside a cell.
The handcuffing model suggests that the rep proteins are doing more than just binding to the DNA. At low plasmid concentrations, when the copy number is low, the iteron-bound rep proteins interact normally with the host cell's replication machinery and the plasmid copies itself.
But when the plasmid concentration is high, a totally different physical interaction takes over.
Right. The proteins bound to the iterons start interacting with each other across different plasmid molecules.
Let's visualize this. Imagine the plasmids are dancers at a crowded party, and the replication machinery is the music.
Okay.
When there are only a few dancers on the floor, they pair up, they follow the rhythm, and replication happens smoothly. But if you pack too many dancers onto the floor, if the plasmid copy number gets too high, the dancers stop pairing up properly. Instead, a dancer reaches out and grabs the hand of another dancer, and then another, until eventually they are all just holding hands in a giant locked circle. They are literally handcuffed together.
And when they are locked in that coupled complex, they become entirely inert. They're physically inaccessible to the host's replication machinery. The music is playing, but nobody can dance.
The physical reality of this is just wild. The sources mention that researchers used electron microscopy to actually look at these pi-mediated interactions. They could visually see the DNA origin fragments looping and physically locking together into these paired structures.
It acts as an absolute, immovable physical roadblock. And what's profound about the handcuffing model is that it relies on direct physical communication between the plasmid molecules themselves to sense their own concentration. It perfectly explains the 10,000-fold protein experiment.
Oh, because adding more protein just makes more handcuffs.
Exactly. If you dump 10,000 times more rep protein into the cell, you aren't triggering replication, you are actually just encouraging more rapid and aggressive handcuffing between the plasmids. The extra protein strengthens the brakes.
So we spent all this time diving into the microscopic turf wars of plasmids, RNA kissing, and handcuffed DNA. Why does this level of mechanistic detail matter outside of a textbook?
Because harnessing these exact mechanisms is the bedrock of the modern biotechnology industry. Scientists have categorized these plasmids into specific incompatibility groups, or INC groups, based on which replication and partitioning systems they use.
And knowing those INC groups is basically the instruction manual for genetic engineering. If you are trying to engineer a bacterium to produce a new biofuel, you might need it to express several complex pathways. You might need one plasmid to produce an enzyme that breaks down plant matter, and a second plasmid to convert that matter into fuel.
But if you are a bioengineer and you don't understand incompatibility, you might accidentally insert two plasmids from the same INC group into your bacterium.
And we know exactly what happens then. The blind cellular machinery forces them into a statistical death match. One plasmid kicks the other out, and your multi-million-dollar biofuel factory fails completely.
Precisely. By intentionally selecting plasmids from completely different INC groups, you ensure that their kissing RNAs and handcuffing proteins don't interact. They ignore each other and stably coexist, doing exactly what you engineered them to do.
The texts also explore using this for environmental protection. You can design bacterial biosensors for bioremediation, like bacteria engineered to glow when they detect heavy metal pollutants in a water supply. You use incompatibility rules to ensure the biosensor doesn't lose its genetic components over time. But my favorite application they mentioned is using this against dangerous bacteria.
the horizontal gene transfer application. It's a fascinating theoretical approach to public health. We know that antibiotic resistance spreads rapidly through bacterial populations in hospitals, precisely because bacteria share these plasmids. They literally hand the blueprints for resistance to one another.
But if scientists were to flood that dangerous bacterial population with a highly stable, harmless dummy plasmid?
A dummy plasmid that is engineered to belong to the exact same incompatibility group as the dangerous antibiotic-resistant plasmid.
Yes. The dummy plasmid infiltrates the bacteria, engages the dangerous plasmid in a turf war for the replication and partitioning machinery, and because the dummy is engineered to be more stable, it outcompetes the dangerous one. You could theoretically cure an entire bacterial population of its antibiotic resistance by weaponizing their own internal incompatibility rules against them.
That's a beautiful concept. But it relies entirely on the premise that bacteria are capable of maintaining absolute perfection in their copying and partitioning mechanisms.
Which brings us to a massive pivot. We have seen how bacteria use incredibly precise physical mechanisms to perfectly control their replication and avoid disastrous errors. They are microscopic masters of quality control. Because for a plasmid, a single unchecked error in copy number means the death of the host and the end of the line.
But what happens when we scale up? When you transition from a single-celled organism maintaining a tiny loop of DNA to a complex organism like a human being tasked with copying billions of base pairs every time a cell divides.
The scale alone suggests that perfection is mathematically impossible. While plasmids play a short, highly regulated game of survival, human evolution is a massive, incredibly messy statistical numbers game. We don't avoid replication errors. Our biology has to find a way to survive them.
Which brings us to the human mutation rate and the fascinating research by Nachman and Crowell on how we actually measure that rate.
The mission of their paper was to figure out exactly how often human DNA mutates per generation. And reading the history of this, it was notoriously difficult to pin down an accurate number.
Historically, it was a near impossible task. Before we had the technology to sequence entire genomes rapidly, geneticists had to rely on indirect methods. One of the most famous attempts was by J.B.S. Haldane in the 1930s.
Right. Haldane didn't have DNA sequencers, so he had to look at what was visible to the naked eye, which were severe disease phenotypes. Specifically, he tracked the incidence of hemophilia.
Hemophilia is a blood clotting disorder, and it's X-linked, which made it slightly easier to track through pedigrees, notably in European royal families. Haldane's method was to look at populations and count how often a child was born with hemophilia when neither parent carried the gene.
By tracking these spontaneous appearances of the disease, he was trying to calculate the underlying error rate of the human genetic copying machine, and he came up with an estimate of about 1 mutation in 100,000 per gene locus per generation.
Which was a brilliant piece of statistical deduction for the 1930s. But as Nachman and Crowell point out in their paper, relying on visible disease phenotypes is a deeply flawed methodology if your goal is to find the true underlying mutation rate of the entire genome.
Because it operates on a massive selection bias, it completely ignores silent mutations. If the DNA polymerase makes a copying error and it changes a DNA letter, but that change happens in a region of DNA that doesn't code for a protein, well, Haldane's method never sees it. Or if the mutation changes a protein, but the new protein functions exactly the same as the old one, there's no disease, no hemophilia. Haldane's method vastly, symptomatically underestimated the true rate because it only counted the catastrophic errors.
Exactly. To find the true rate of a ticking clock, you cannot look at a mechanism that is constantly being reset by natural selection. You need to find a part of the genome that is completely free from evolutionary pressure.
You need a piece of DNA that literally doesn't do anything. Because if it has no function, mutations can just pile up randomly, generation after generation, without harming or helping the organism. It acts as a perfect, undisturbed metronome.
This relies on the neutral theory of molecular evolution. And the specific type of DNA sequence the researchers utilize to act as this metronome are called processed pseudogenes.
The concept of a processed pseudogene is like a piece of evolutionary archaeology. Basically, during the normal operation of a cell, a functional gene is transcribed into messenger RNA so it can be translated into a protein. But occasionally, the cell makes a bizarre mistake. An enzyme reverse transcribes that mRNA back into a solid piece of DNA, and the cell accidentally shoves it back into the main genome at a random location.
But crucially, because it went through the mRNA phase, it has been stripped of all its regulatory elements.
Right. I picture it like taking a photocopy of a cake recipe from a cookbook, but your photocopy cuts off the title, the oven temperature, and the instructions on how to mix it. And then you just glue that useless list of ingredients into the middle of a dictionary. It's dead on arrival, has no promoter to turn it on, and it has no introns. It is a completely useless stretch of code.
And because it is dead code, natural selection is blind to it. Whether it mutates or stays pristine, the human organism functions identically. So over millions of years, random copying errors accumulate in this sequence at a steady, uninterrupted rate.
The researchers in this paper took 18 of these processed pseudogenes, 12 located on the autosomes and 6 on the X chromosome, and they compared the human sequence directly to the chimpanzee sequence.
But when you evaluate a study like this, you have to ask a critical methodological question. If pseudogenes are essentially dead genes accumulating random garbage for millions of years, how do the researchers know they're looking at the exact same dead gene in a human and a chimp?
Yes. How do you prove that this isn't just a similar-looking piece of random noise?
They had to guarantee a concept called orthology. Orthologous means that the pseudogene found in the human genome and the pseudogene found in the chimpanzee genome are derived from the exact same singular ancestral integration event.
To prove that, they couldn't just sequence the pseudogene itself. They had to sequence the DNA immediately surrounding it, the flanking regions.
Exactly. They looked at the integration sites. If you find a dead pseudogene in a human, and you find a nearly identical pseudogene in a chimpanzee, and in both species, it is glued into the exact same location, surrounded by the exact same sequence of functional DNA.
It proves beyond a shadow of a doubt that the accidental gluing event happened before the human and chimpanzee lineages split from their common ancestor, which evolutionary biologists estimate was around 5 to 6 million years ago.
It guarantees that they're looking at the exact same ticking molecular clock in both species.
Okay, so they have their perfect, undisturbed molecular clocks, 18 of them, covering over 16,000 base pairs of DNA. What did the math actually reveal?
Across those 16,000 base pairs, by carefully aligning the human and chimpanzee sequences, they found 199 distinct mutational differences.
199 differences out of 16,000 letters. When they ran the calculations on the autosomal divergence, the non-sex chromosomes, it came out to about 1.33%.
And from that 1.33% divergence, utilizing established estimates of ancestral population sizes and an assumed average generation time of roughly 20 years, they were able to calculate the underlying baseline mutation rate of primate DNA.
The number they arrived at is approximately 2.5 times 10 to the negative 8 mutations per nucleotide site per generation.
Now, in scientific notation, that looks like a remarkably small number, but you have to scale it up to the reality of human biology.
Let's translate that into numbers that actually mean something for a human body. The human genome is diploid. We inherit one complete set of chromosomes from our mother and one from our father. In total, that is roughly 7 billion base pairs of DNA. If you multiply 7 billion by that mutation rate of 2.5 times 10 to the negative 8,
you arrive at an average of 175.
175 new mutations per generation. Every single time a human child is born, they are carrying, on average, 175 brand new genetic mutations that absolutely did not exist in either of their parents' genomes. 175 times a copying machine made a mistake.
When you compare that to the absolute lethal perfection demanded by the bacterial plasmids we discussed earlier, it is a staggering amount of genetic noise to introduce into a system in a single generation.
But the paper goes further, pointing out that these 175 mutations aren't just evenly distributed across the genome like a fine mist. There is massive mutational heterogeneity. There are specific hotspots where the DNA is highly vulnerable.
The most prominent hotspots they identified are known as CpG dinucleotides.
A CpG site is very simple structurally. It's just a cytosine nucleotide, a C immediately followed by a guanine nucleotide, a G, on the same DNA strand. Why are they so vulnerable?
The vulnerability lies in the chemistry of how mammals regulate their genes. In mammalian genomes, these specific cytosines are very frequently methylated. An enzyme physically attaches a chemical methyl group to the cytosine molecule, usually as a way to silence or turn off nearby genes.
But attaching that methyl group fundamentally destabilizes the cytosine molecule.
Highly destabilizes it. The 5-methylcytosine undergoes a spontaneous chemical reaction called deamination.
Spontaneous deamination. The cytosine literally just loses an amino group to the surrounding water in the cell, and when it loses that piece, its chemical structure transforms. It is no longer cytosine, it becomes a thymine.
It physically changes the letter of the genetic code from a C to a T. And this is incredibly insidious because thymine is a normal, naturally occurring base in DNA.
Right. If the DNA is damaged in a bizarre way, the cell's repair machinery usually spots it immediately and cuts it out. But here, the repair machinery scans the DNA, sees a T, and says, well, thymine belongs in DNA, looks fine to me. It often fails to recognize that an error has occurred until a cell divides and the mutation is permanently locked in.
The data in the paper reflects this chemical vulnerability perfectly. They found that transition rates at these specific CPG sites are an order of magnitude higher, 10 times higher than the background mutation rate at other sites in the pseudogenes.
10 times higher. It's a massive, glaring vulnerability in the chemical stability of our genetic code. The researchers also noted something interesting about the types of errors. Single nucleotide substitutions where just one letter is swapped for another, like the C to T we just talked about, were 10 times more frequent than length mutations.
Length mutations being insertions or deletions where the polymerase accidentally skips a letter or adds an extra one.
Which logically makes sense. An insertion or deletion usually causes what's called a frame shift. Because DNA is read in 3-letter words, if you add one extra letter, it shifts the entire reading frame for the rest of the gene, scrambling everything downstream into gibberish. Frame shifts are far more likely to be fatally destructive, so it makes sense that the basic biological machinery is at least slightly more careful about avoiding them compared to simple typos.
The researchers also utilized their data to test a very long-standing, somewhat controversial theory in evolutionary biology regarding where these mutations originate, which is male-driven evolution.
The hypothesis being that the vast majority of these 175 new mutations originate specifically in the male germline.
To test this, they compared the divergence rates of the pseudogenes located on the autosomes against the pseudogenes located on the X chromosome. They calculated the ratio of the male mutation rate to the female mutation rate, which is represented mathematically by the Greek letter alpha.
And based on their data from human and chimp sequences, they estimated alpha at 3.6. So males mutate roughly 3.6 times more rapidly than females. Why is that? Does the male body just have worse DNA polymerase?
No, the copying machinery is identical. The difference lies entirely in the cellular life cycle. The male germline, the biological process of spermatogenesis, goes through significantly more continuous cell divisions throughout a male's lifetime compared to the female germline.
Oochogenesis in females is largely completed in terms of cell divisions before the female is even born. But spermatogenesis is a continuous ongoing process of cell division from puberty onward.
Exactly. More divisions mean more times the DNA has to be unzipped and copied, and every single time it is copied, there is a statistical chance for the polymerase to make an error. It's purely a numbers game.
So how does the X chromosome prove this?
Think about where the X chromosome spends its time. In a given population, females have 2 X chromosomes and males have 1. This means that statistically, any given X chromosome spends 2/3 of its evolutionary history inside female bodies, and only 1/3 inside male bodies.
Because it spends twice as much time in the female germline, it is physically exposed to far fewer rounds of replication-heavy cell divisions than the autosomes, which spend equal time in both sexes.
Precisely. Because it avoids the male germline more often, the X chromosome accumulates mutations more slowly. The calculation of alpha equal to 3.6 perfectly supports this male-driven mutation theory. And importantly, the researchers concluded that this provides no evidence that the X chromosome inherently evolved a lower mutation rate for some special adaptive reason. It isn't shielded by magic. It's shielded by simple mathematics regarding sperm versus egg production.
Which brings us to the final and undoubtedly the most provocative, mind-bending part of this entire deep dive. We have established that every single human is born with roughly 175 new genetic mutations.
The unavoidable question is, how many of those 175 mutations are actually harmful?
To figure this out, the researchers had to estimate what percentage of the massive human genome is actually under constraint, meaning what percentage actually does something critical where changing a letter would break a necessary function.
They use an incredibly conservative estimate to avoid overstating the problem. They only look at coding sequences, the specific stretches of DNA that directly code for proteins. That makes up only about 1.7% of the total human genome.
So if we assume that only 1.7% of the genome is critical, and we know we have 175 new mutations per generation, you just multiply 175 by 0.017.
And that calculation gives you the genomic deleterious mutation rate, commonly denoted in population genetics as U. U is approximately equal to 3.
U equals 3. Let that sink in. This means that every single human being on Earth is born with at least 3 brand new, fundamentally harmful mutations. 3 pieces of their critical protein-coding genetic code have been broken compared to their parents.
And remember, because they only counted the 1.7% coding region, this is highly conservative. If the vast non-coding regulatory regions, the switches that turn genes on and off, are also under constraint, the true value of U could be significantly higher.
But even at the conservative estimate of U equals 3, we hit a massive biological paradox.
The paradox of multiplicative fitness effects.
Okay, let's unpack this for you, the listener, because this is where the math gets terrifying. If mutations have multiplicative fitness effects, it means they act completely independently of one another, and each new mutation reduces your overall evolutionary fitness by a certain set percentage, and those percentages compound over generations.
If the deleterious mutation rate is 3 per generation, and they compound multiplicatively, population geneticists have calculated that the mathematical reduction in average fitness for the species drops to roughly 0.05.
To put a fitness of 0.05 into perspective, in order to simply maintain a constant population size, not even grow, just replace the current population a human female would need to give birth to 40 children.
40 children per female, just to ensure that 2 of them survive the crushing weight of their genetic errors and reach reproductive age.
Obviously, human women are not birthing 40 children on average. Human biology cannot support that.
Not even close.
So here is the ultimate paradox. How are we not all dead? How has the human race not gone completely extinct under the compounding weight of 3 new harmful mutations per person per generation?
To resolve this massive mathematical paradox, the authors point to a concept called synergistic epistasis, which is sometimes referred to in the literature as truncation selection.
Synergistic epistasis. It sounds incredibly complex, but the underlying concept is actually very intuitive.
It essentially argues that harmful mutations do not act independently. They don't just multiply quietly in the background. They interact with each other.
I want to use an analogy here. Imagine your entire genome is a precarious Jenga tower. Pulling out one block from the tower that represents one deleterious mutation. The tower wobbles slightly, but it's fine. It stands. You pull out a second block, a second mutation, still fine. You can carry 1, 2, or even several of these genetic mutations with minimal effect on your daily life or your reproductive fitness.
Because the biological systems have redundancies.
Exactly. But once you cross a certain threshold,
that 3rd or 4th or 5th block you pull out doesn't just lower your fitness by a set 2%. It interacts synergistically with the structural weakness of all the other missing blocks, and it causes the entire tower to collapse simultaneously.
In biological terms, what does that collapse look like?
It means that once an individual accumulates a specific threshold number of mutations, the combined synergistic effect is lethal, or at least completely sterilizing. That specific individual does not survive to reproduce, and when they fail to reproduce, all of those accumulated mutations, the entire precarious load of missing Jenga blocks, are simultaneously purged from the human gene pool in one single event.
It's nature's incredibly brutal way of clearing the cache. We survive as a species because the mutations stay relatively hidden in the Jenga tower, passed down quietly, until a specific individual's tower finally collapses, taking all that bad code out of circulation at once.
It is a harsh reality of population genetics, but it is the only mathematical way to reconcile a staggering mutation rate of 175 errors per generation with the continued survival and relatively low birth rate of the human species.
It is truly staggering to think about the journey we've taken today. We started by looking at bacteria. We saw how plasmids use incredibly precise physical mechanisms like the RNA kissing complex that intercepts primers and the Iteron hand-tuffing model that physically locks DNA in place to perfectly regulate their copy numbers.
Because in their microscopic world, there is no room for a genetic load. If a plasmid replicates too much or fails to partition, it is instantly and ruthlessly destroyed. Their environment is immediate and unforgiving.
Then we zoomed out millions of years. We saw that human biology hasn't avoided these replication errors, we actually embrace them. The mechanisms of human life are messy, the polymerase proteins make typos, and our CpG sites spontaneously decay into thymine. We accumulate 175 of these errors every single generation.
And yet, through the brutally efficient mathematics of synergistic epistasis, the human lineage doesn't just continue, it adapts, it evolves.
Which brings me to a final thought for you to mull over today as you go about your life. Based on this concept of synergistic epistasis, you, the person listening to this conversation right now, are walking around carrying a heavy, silent load of genetic mutations passed down from thousands of ancestors, plus 3 brand new ones of your very own. You are a wobbling Jenga tower. You are alive and breathing and thriving only because your specific, unique combination of errors hasn't yet crossed that invisible threshold of collapse. Every single new generation is a massive genetic gamble, a roll of the molecular dice. And yet, against all the mathematical odds of the genetic load, against the constant ticking decay of your DNA, here you are, perfectly capable of learning, understanding, and exploring the universe.
It truly puts both the immense fragility and the astonishing resilience of life into a brilliant perspective.
Thank you for joining us on this exploration. Until next time, keep questioning the simple diagnoses and keep looking for the invisible rules.